Hello and welcome to today's lesson on nucleotides and nucleic acids from A-Level Biology. This presentation is more structured around the OCR A-Level specification but has been adapted a bit to try and suit as many specifications as possible. An important part of this topic is understanding the structure of a nucleotide. So nucleotides bond to form nucleic acids, and there are two main nucleic acids which are DNA and RNA. Now nucleotides are essentially a monomer of nucleic acids. There are 5 possible nitrogenous bases that nucleotides can have, which are adenine, guanine, cytosine, thymine, cytosine, uracil, but uracil is only found in RNA. You don't really need to know this, but that's primarily to do with its stability, which doesn't really affect RNA. They have complementary base pairings, so guanine and cytosine will always bond together, and adenine and thymine will always bond together as well. Now, the picture on the left is more what you would have seen at GCSE level and perhaps had to draw as a nucleotide. Now, this is still perfectly okay to follow at A level, but it is kind of important that you understand more of the general structure. So a nucleotide will have a phosphate group, a pentose sugar, and then a nitrogenous base. The nitrogenous bases are what we mentioned above. The pentose sugar is what changes between DNA and RNA, so in RNA it's ribose, whereas in DNA it's deoxyribose. And then the phosphate group, there can be one or more, and when there are more, that's when you get different compounds like ATP, for example. So when discussing nitrogenous bases, we can categorize them as either a purine or a pyrimidine. A purine is two-ringed, so for example adenine and guanine, whereas pyrimidines are single-ringed. Now, these nitrogenous bases in DNA form hydrogen bonds with each other. When we think back to the biological molecules topic, we should know how hydrogen bonds are formed. If not, that will be coming up in a later lesson. But so essentially they form hydrogen bonds with each other only in accordance with their complementary base pairs. As such, a purine will always bond with a pyrimidine. Another important point to note is that adenine and thymine will hydrogen bond with 2 bonds between them, and guanine and cytosine will hydrogen bond with 3 hydrogen bonds between them. Now, the relevance of this is that the difference in 2 and 3 hydrogen bonds is what causes the spiral structure of DNA, which you would have seen. So, as mentioned, a nucleotide can have more than one phosphate group. Now, when there are multiple phosphate groups, the nucleotide is called a phosphorylated nucleotide. The phosphate group will always be bonded to the pentose sugar in the nucleotide, and this is then bonded to the nitrogenous base. So in ATP and ADP, which is a good example, the phosphate group is bonded to a ribose sugar, and then this is bonded to adenine. Now we see that when there's 2 phosphate groups, that's ADP, and when there's 3 phosphate groups, that's ATP. This is because ATP's full form is adenosine triphosphate, so that obviously meaning 3 phosphate, because it's 3 phosphate groups, and ADP would be adenosine diphosphate, because again, 2 phosphate groups. It's very important that we understand how nucleic acids are formed. So first of all, nucleic acids are essentially a macromolecule that are adapted to storing genetic information and have the ability to be used to transfer that genetic information. They're formed from nucleotides, which are again the monomers, being bonded via phosphodiester bonds. These are formed in a condensation reaction, which therefore means that water is produced as a byproduct with one water molecule being produced for each bond that is formed. And these phosphodiester bonds form the backbone of the nucleic acid strands. When a nucleic acid is broken down, a hydrolysis reaction is used in which a water molecule is essentially used to break the phosphodiester bond, which is pretty much just a reverse reaction of the condensation reaction. This diagram essentially just illustrates how a condensation reaction works to form a phosphodiester bond, and what it's important to point out is that the phosphodiester bond is formed between the phosphate group and the given sugar, and again we can see the way in which the water molecule is produced. You can be asked to draw out a basic reaction scheme for this, so having a diagram akin to this kind of memorized or knowing it in a degree of detail is important, but you don't necessarily need to know the exact structure of the phosphate or each component in the nucleotide. So the two types of nucleic acid are DNA and RNA, with DNA being deoxyribose nucleic acid and RNA being ribonucleic acid. The difference in naming structure refers to the variation of the pentose sugar in the nucleotide monomer of DNA and RNA. So when we look at the variation in the pentose sugar, we see that deoxyribose has essentially one less oxygen on that, you should never describe it like this, but essentially that bottom right carbon, which is again shown in the diagram. Equally, it's important to note that thymine, which is a base in DNA, is replaced by uracil in RNA, as I mentioned before. As a result of this, in RNA, uracil will bind to adenine as opposed to thymine. So getting on to the nature of genetic code, we have to understand firstly that DNA is structured such that a triplet, which is a series of 3 bases, is known as a codon, and that codon will code for a given specific amino acid. Now, a gene is essentially a series of triplets or codons, which each code for an amino acid. Thus, a gene codes for a specific sequence of amino acids, which then form polypeptide chains with peptide bonds between the amino acids. In DNA, there are also coding regions and non-coding regions. Now, the coding regions are what we would expect, coding for the sequence of amino acid, but the non-coding regions are related to gene expression. This comes up in far more detail in a later topic, and we will go through that. But for now, just know that they can be related to gene expression. Genetic code also has some specific features. So first and foremost, it's universal. So this means that in almost all living organisms, the same triplet or codon will code for the same amino acid. Now, DNA is also degenerate, and genetic code is also degenerate. So what that means is all amino acids, other than 2 specific ones, which I'm not sure if you have to know, but they are methionine and tryptophan, have more than one triplet which codes for them. The importance of this is that the effect of a point mutation is somewhat decreased because there's still a chance that the same amino acid is coded for, thus the protein is still functional. Genetic code is also non-overlapping, so it has to be read from a fixed point, and if a base is added or deleted, for example, there will be what's called a frame shift. Again, this is gone into in far more detail in later topics, which affects all subsequent bases, and the code for all amino acids after that point is changed. Now, it's very important we understand the process of semi-conservative DNA replication. Now, it's called semi-conservative as one of the strands of DNA in each of the new copies is preserved from the old DNA strand or original DNA strand. The process itself is relatively simple. The DNA double helix will be unwound and unzipped. Now, the unwinding utilizes the gyrase enzyme. You may not need to know that based on your specification. The DNA will then be unzipped using DNA helicase. Now, what DNA helicase does is it essentially catalyzes the process of the hydrogen bonds between the complementary base pairs being broken. This obviously exposes the bases that were previously bonded to their complementary bases, and thus free phosphorylated nucleotides will hydrogen bond to the exposed bases in accordance with the complementary base pair rule. DNA polymerase will catalyze the formation of phosphodiester bonds between the nucleotides in the 5-3 direction. The reason the direction is important is because it only functions in the 5-3 direction, a leading strand and a lagging strand will form, with the leading strand being the strand that runs entirely in the 5-3 direction and the lagging strand running in the opposite direction. And so on the lagging strand, the formation of phosphodiester bonds has to be catalyzed in fragments such that they're then joined by ligase enzymes. These fragments are called Okazaki fragments, and you may or may not need to know that again based on your specification. Now, the process of DNA replication will occur across the entire DNA strand, and both individual strand, so to speak. And then when the entire strand is done, both individual strands have been used as templates, and the result is 2 identical strands. So using the diagram on the left, we can kind of visualize the process of semi-conservative DNA replication. So we can see that each of the new strands are firstly identical, which we can't necessarily see in this diagram, but they are identical, and each strand has half new synthesized DNA and half old or original DNA from the strand, from the original strand. Now, on the diagram on the right, we can see kind of a more detailed process diagram of DNA replication. Now, you don't need to know everything on the screen, as it's a bit more of a high-level diagram, but what I wanted to discuss was the presence of an RNA primer. Now, an RNA primer is a short segment of RNA that acts as a binding site for DNA polymerase to initiate the catalysis of the formation of phosphodiester bonds, which is important for DNA replication. Now, the reason this primer is needed is because DNA polymerase can only add nucleotides to an existing chain. It can't, start the chain itself. So the primer is needed to enable the DNA polymerase to act in the first place. We can also see that on the strand which runs in the 3-5 direction, as opposed to the 5-3 direction, which DNA polymerase can act in, there are Okazaki fragments, which are then linked by DNA ligase. We see DNA ligase acting there, and that's just a good diagram to enable you to kind of visualize what happens. Now, never write this in an exam, but a way you can kind of think about it is DNA polymerase acts in the 5-3 direction, and DNA helicase will be kind of catalyzing the process of breaking those hydrogen bonds all the way along the DNA strand. Now, DNA polymerase can kind of follow the DNA helicase all the way, catalyzing the process of phosphodiester bond formation. But on the lagging strand, there isn't enough room, essentially, because not enough hydrogen bonds have been broken for it to catalyze in one clear sequence. Therefore, it essentially has to go back and catalyze it in segments, and then those segments have to be joined using DNA ligase. Now, before moving on to protein synthesis, it's important that we understand the types of RNA. So there's mRNA, which essentially acts as a vector of sorts between the genetic information written in the nucleic acids in the nucleus, so DNA, and the ribosome tRNA. tRNA serves as an adapter of sorts between the genetic instructions and the protein products. So it kind of brings the two together, if that makes sense. And rRNA is ribosomal RNA. This essentially combines with proteins and enzymes in the cytoplasm to form ribosomes, which obviously act as the site of protein synthesis. So what is protein synthesis? It's essentially the process by which a protein is made from genetic code. There are 2 key stages, which we'll explore in far more detail now: transcription and translation. So the process of transcription. So the DNA will be unwound and unzipped, which is pretty much the same as with replication. Free-floating RNA nucleotides will then bind to the complementary nucleotides through hydrogen bonding, and remember, uracil will bind with adenine instead of thymine. These free-floating RNA nucleotides are essentially found in the nucleus, the same as the DNA nucleotides, and again will obviously bind in accordance with the complementary base pair rule. Now, RNA polymerase catalyzes the formation of phosphodiester bonds between the nucleotides, forming an mRNA strand. The hydrogen bonds that are formed are extremely temporary, and so those break the minute the mRNA strand is formed. This will only occur on one strand, which is the template strand. The other strand is known as the coding strand, and the coding strand is the strand which the RNA essentially is the same as. Again, don't write that in an exam, but that's a good way to think about it. And obviously it's going to be the same with the exception of uracil being there instead of thymine. Once the mRNA strand is formed, the mRNA will pass out of the nucleus. This occurs via nuclear pores, and it will then be in the cytoplasm, where the next stage of protein synthesis will take place. Now, towards the bottom of this diagram, we can see that the RNA essentially follows the same sequence as the coding strand, as I mentioned before, but is complementary to the template strand. We can also see the essential process by which the mRNA would be made. So there's the unzipping, and then the complementary RNA nucleotides will bond, forming the mRNA strand after the catalysis of phosphodiester bonds by RNA polymerase. So now on to the process of translation. This will occur in the cytoplasm of the cell. The mRNA, after leaving the nucleus, will either bind to a ribosome that's free-floating or on the RER, the rough endoplasmic reticulum. The cytoplasm has free-floating tRNA and amino acids. The tRNA has specific anticodons, which is essentially a triplet that will be complementary to a given codon. So the tRNA will then bind with its specific amino acid, and the anticodon will then bind, according to the complementary base pair rule, with its complementary triplet or the codon on the mRNA. There are two tRNA molecules that will bind at the same time in the ribosome, and as each one will have its specific amino acid attached, the two amino acids are brought obviously quite close together, which enables the formation of a peptide bond between those two amino acids. Now, the formation of a peptide bond requires ATP energy, and that ATP is provided by the mitochondria in the cell. Now, this process of the codons being read using the anticodons of the tRNA will occur across the entire mRNA strand until a stop codon is read. This produces a polypeptide chain, which will then fold via the rules of primary, secondary, tertiary, and quaternary protein structure into a protein. The resultant polypeptide chain obviously has a specific sequence of amino acids, that's the primary structure of a protein, which matches essentially the genetic code from which it was transcribed and translated, and therefore you have a specific protein formed due to that specific sequence of amino acids. And that is pretty much the conclusion of the majority of the talking points of this topic. So you should have a fairly good understanding of the nucleotides and nucleic acids topic now. If you need any more help or anything more specific, I do offer tuition sessions, one-to-one, and it can be in a group if that's what you'd prefer. I also offer a ton of free notes on my website that's linked in the description. If you're interested in tuition, you can either comment below, or via my website there's a contact link, and we can arrange that. In one-to-one tuition sessions, we'll cover all of this stuff in far more detail. We'll go over exam technique. We'll use an exam technique guide that I've created by answering over 10,000 marks worth of past paper questions and doing countless hours of revision myself. And then we'll put that into practice by getting you to answer questions, and also really having a deeper discussion about any of the points you're struggling with. So yeah, I hope you found the lesson useful. We'll be covering another lesson next week. I'm aiming to get a lesson out every week. So for anything more specific, please contact me either in the comments or by the website linked in my description. And yeah, see you next week.